Hello dear students, welcome into the first lecture on high performance thin layer chromatography. Myself, Mr. M.S. Bale, I am working as an assistant professor at Gokhale Education Society's Arts, Commerce and Science College, Jawar. So, we have completed high performance liquid chromatography in previous lecture. So today we are going to learn introduction and principle of high performance thin layer chromatography, that is HPTLC. So introduction and principle of this high performance thin layer chromatography. Actually, all of you know what is TLC, that is thin layer chromatography. This technique actually all of you learn in previous class in detail. So actually theory of the TLC and HPTLC will be the same, just some of the part in HPTLC will be changed. So in HPTLC here, this most of the part will be done by using the instruments, and we are getting the results in few minutes. That means our reading will be obtained in very fast as compared to the previous TLC technique. So HPTLC is a versatile separation technique and is official is in most of the pharma copies for determining content uniformity, purity profiles, assay values and dissolution rates in ultimated number of monographs. It is precisely for the reason that almost every laboratory today is equipped with HPLC system. See, for our present study, we have chose this HPTLC as a method of analysis. That means this HPTLC we are using for the analysis purpose. In homeopathic formulations, no standardization method are developed on the basis of fingerprinting characterization. So our aim is to be develop a standard procedure for evaluation of homeopathic formulations. So actually these are the various area where we can use this HPTLC technique for the analysis purpose. So in this case, HPTLC is the most simple separation technique available for the analyst. It can simultaneously handle several samples, even different nature and composition. The theory of TLC and HPTLC is exactly the same. The high performance is the result. Three things: use of pre-coated plates with uniform thickness and small particle size of the adsorbent. See, that means in HPTLC, here we are using pre-coated plate with uniform thickness. Generally in TLC we are using a glass plate and the silica slurry, where the glass plate we are absorbing that silica slurry on that glass plate. That will be the ordinary method, or you can say general method. But in HPTLC, here we are using pre-coated plate rather than that ordinary plate, or you can say glass plate. Sample application is done very uniform and accurately with an automated applicator. See, in HPLC, actually the machine will be used, or you can say instrument will be used to give the spot, or you can say apply the sample on pre-coated plate, but in TLC we are using small capillary for the spotting of the sample material. That will be TLC will be the very tedious and time consuming, but in this HPLC the instrument will be done in few minutes. This sample application. Detection of separated components done using scanning instruments. Actually, whatever spot where we gave in TLC, in case of TLC, that spot we are separating in saturation chamber, separated spot we are detect by separated color spot. That will be the very time consuming one, but in HPTLC that separation, or you can say separated components, we are directly scanning with the help of the instruments, so it will be very fast as compared to the TLC, ordinary TLC method. So that's why we called as high performance thin layer chromatography. So now we will see the steps involved in HPTLC. What are the steps when we do the HPTLC technique in sample analysis? For that, what step we have to follow, that we are going to see now. See, first step is the selection of HPTLC plates and solvent. That means what type of the HPTLC plate we have to select and what type of solvent will be used for the making the mobile phase. So pre-coated TLC, or can say HPTLC plates, which is having size 20 by 20 centimeter, or we can say 10 by 10 centimeter, depends whatever requirement of our analysis. According to that, we have to select size, that is 20 by 20 or 10 by 10 centimeter, with aluminum or polyester support, that will be usually used for the sample application. These plates can be cut to size and shape format to suit particular analysis by using general purpose of scissors. That means with the help of scissors or blades, we can cut that plate according to our requirement, and that plate will be used for the applying sample. But before application of the sample, we have to activate that pre-coated plate. Actually, activation means what? Some contamination may be present on that pre-coated plate, either humidity, moisture, or another contamination is there. For that purpose, we have to activate that pre-coated plate. Actually, freshly opened box of TLC or HPLC plates usually does not require any activation, because that will be the fresh one, there is no contamination. So actually, suppose plate exposed to high humidity or kept on hand for long time maybe have to be activated by placing in oven at 110 to 120 degrees Celsius for 30 minutes prior to sample spotting. That means before spotting the sample on that TLC plate, suppose we have to remove humidity or some contamination on that, we have to heat that plate in oven at 110 to 120 degrees Celsius temperature for 30 minutes. Then that plate we can use to apply the sample. So actually this is the first step. Actually, second one will be the sample preparation. All of you know sample preparation. Actually, sample preparation and standard preparation is an important tool in HPTLC. Samples should be prepared as solution form with suitable solvent in proper concentration and used for analysis. That means, first of all, we have to check the solubility of the sample into the solvent. According to that, we have to select the mobile phase, or you can say solvent. The sample preparation is not as demanding as for other chromatography techniques. However, several steps for sample pre-treatment may be necessary, such as sampling, mechanically crushing, extraction, filtration, and enrichment of the minor compounds. Proper sample preparation is an important prerequisite for success of thin layer chromatography separation. After checking the sample dissolution, or can say miscibility into the solvent, then we can apply that sample on that TLC plate. That will be the, I think, third step. See, application of sample, that is the third step. Actually, sample apply on the TLC plate, there is two types, as a band or as a spot. Spot means circular spot that were done in TLC also with the help of that capillary. So likewise we can apply the spot, circular spot on that TLC plate with the help of the nanomat machine. This nanomat, it is actually the device which is used to give the spot on TLC plate. Okay, and the second will be the as a band. For as a band, we have to use linomat machine. Linomat, it is also again the device which is used to give this band on TLC plate. So these are the actually two types of application of sample. Now fourth step will be the development, that means separation. After spotting the sample on TLC plate, we have to develop, or you can say we have to separate the sample from that original spot, or you can say original band, for checking purpose. So for that we have to choose a chamber. Into the chamber we have to keep our mobile phase. After saturation of that chamber, we have to place our TLC plate, spotted TLC plate, into that in vertical position and separate that sample component. After separation of the sample component, we have to detect, that means the fifth number step will be the detection, including post chromatographic derivatization, that means identification of spots. In general TLC, we use a iodine chamber to identification of spot, but in this case we are using actually for the detection purpose the densitometer, that I will be either single beam or double beam densitometer, that will be used for the identification of the spot on TLC plate. Sixth number step will be the quantization. After identification of spot, we have to quantize it. Then at the last part of these steps, that is documentation. After detection of the spot, we are getting the chromatogram, just like HPLC. We are getting chromatogram into our monitor, because this HPLC is carried out, is totally automated. In monitor we are getting the chromatogram. That chromatogram we have to save or take a printout of that graph, or you can say chromatogram. That will be the documentation part. Actually these are the steps to follow while analyzing any kind of sample with the help of this HPTS technique. So actually the sequence of steps in operation of HPLC. See this is the sequence that we have to follow. Actually, this is the schematic procedure of HPLC. These steps we have already seen. See, sample and standard preparation, when we are doing at that time, we have to make ready selection of chromatographic layer pre-washing, layer pre-conditioning. Then we have to apply the sample and standard. For the comparison purpose, we have to apply along with sample. Near spot will be the standard. Then chromatographic development, that means separation, detection of spots, scanning and documentation of chromatoplate. These are the actually steps to follow. So this is actually introduction and principle of the HPTLC. So in the next lecture, we are going to see actual all these steps in very detailed manner. So thank you for today.